So for this session, we'll take a look at acid-base regulation of the renal system. Hopefully, at the end of the session, we'll be able to discuss the basic principles of acid-base physiology, including the role of the bicarbonate carbon dioxide buffer system, describe how the kidney maintains acid-base homeostasis by adjusting the rate of net acids excretion to equal the daily acid load, enumerate the characteristics of the different acid-base disorders and the compensatory responses they initiate. Lastly, to explain the factors responsible for generation and continuation of metabolic alkalosis and acidosis. So for starters, let us just review definitions of acids and bases. So from your undergraduate years, you know that acid are defined as substances that can donate hydrogen ions, or some definitions say that can donate hydrogen, aside from hydrogen ions, they can donate protons. No? On the other hand, your base are substances that can accept hydrogen ions or they can uh, uh, accept protons. So these are the things that are associated with the definition of acids and bases. So generally, you can classify your acids into two categories. One could be your carbonic acid, the other is your non-carbonic acid. So the difference is that the carbonic acid generally associated with volatile acids or with carbon dioxide, and usually the body produces about 15,000 millimole per day, and most of these are eliminated through the lungs. Compared to carbonic acid, there are just few non-carbonic acids in terms of volume that are produced by the body, or you call them your non-volatile acids, such as your phosphoric acids and sulfuric acids. And the body usually produces about 50 to 100 milliequivalent per day. And they're usually products of protein metabolism. So usually they are eliminated by a combination with buffers in the body and subsequently it's excreted by the kidney. So examples of your acids will be your carbonic acid, your hydrochloric acid, or your ammonium, or your dibasic phosphates. On the other hand, we could also have uh, other acids and bases in the body, but these are the most common ones that you can encounter in the clinics. So the complete metabolism of your carbohydrates and fats in the diet will require oxygen and insulin. And usually this metabolic breakdown of food will yield carbon dioxide and water. So when the lungs function normally, it's able to produce carbon dioxide and it's able to excrete it. It's eliminated. So the 20 moles per day of carbon dioxide produced uh, generally has no impact on the systemic acid-base balance because of the following reactions. So take note of uh, this important reaction here. So you have your carbon dioxide produced by the breakdown of your carbohydrates and fats plus water in the bloodstream. So in aqueous solution, when it's dissolved in aqueous solution, your carbon dioxide can combine with water, H2O, produce your uh, bicarbonate. So this carbonic acid can be broken down into your bicarbonate and your hydrogen ion. So these are mass equations, meaning to say that the rate of the conversion from one form to another is dependent on the presence of substances in the body. So if you have an excess of carbon dioxide in the body, that will this reaction will move to the right. So you will produce more carbonic acid and bicarbonates, hydrogen ion. Sorry, it's called. If you have more hydrogen ion, the equation will shift to the left, and as a result, you'll eliminate more carbon dioxide. So uh, alterations in ventilation by changing the partial pressure of carbon dioxide in the blood will alter your pH. So an increase in your partial pressure of carbon dioxide will produce acidosis because it will move the equation to the right, so producing more hydrogen ion. So, however, a decrease in carbon dioxide will shift the equation to the left so that it will produce alkalosis because there will be a reduction of your hydrogen ion. So when your body metabolizes amino acids in the protein, so remember your protein metabolism will produce your amino acids. They can produce either acids or alkali or bases depending on the specific amino acid. However, the metabolism of dietary protein usually produce net acids, so either hydrochloride or uh, sulfuric acid. So these acids are often referred to as non-volatile acids, and generally they are rapidly buffered. So they are buffered by in the body. So uh, the body is a way of compensating for an increase of your non-volatile or non-carbonic acid. So each every day, so as I mentioned earlier, the breakdown of your food dietary consumption, so like your fat and carbohydrates, will result in the generation of approximately 15,000 millimole of carbon dioxide. So although technically carbon dioxide is not an acid, it can combine with water to form your carbonic acid. So thus there will be progressive accumulation of acid if the endogenously produced carbon dioxide were not excreted. So if your carbon dioxide produced by your metabolism of your food products is not excreted, then there will be an accumulation of acid in the body. So this is prevented by the loss of carbon dioxide via your lungs, no? So through the lungs via alveolar ventilation, so that the body is able to deal with this 15,000 millimole of carbon dioxide as a byproduct of your metabolic activities in the body through pulmonary ventilation. On the other hand, your protein products, no, that produces non-carbonic acids are primarily generated from the uh, breakdown of your proteins. So in particular, the oxidation of sulfur-containing amino acids results in the formation of your sulfuric acid. So in comparison to the very high rate of carbon dioxide production, which is about 15,000 millimole per day, only about 50 to 100 mex, no, of non-carbonic acid is produced each day. So how does the body eliminate this acid uh, this net acid, indigenous acid production of the body, or NA. So usually, your net indigenous acid production in the body uh, is eliminated by a two-step process. First, there's an initial combination with extracellular bicarbonate. So you have your extracellular bicarbonate and intracellular buffers to minimize the change in free hydrogen concentration, because uh, your normal hydrogen concentration, hydrogen ion concentration, is limited to a very narrow range, no, of values. And uh, in order for this accumulation of acids in the body not to swing this hydrogen ion concentration wildly, the buffers in the body act to stabilize, no, the presence of these excess acids in the body. And so once the buffers have stabilized these acids, then subsequently these acids are excreted, no, by the kidney through the urinary tract. So one of the things that we should take a look at with regards to your buffering system is to see what part of the renal uh, system each of these buffers are operative. So let's take a look at your bicarbonate, which are one of your primary buffers, no, in the body. So bicarbonate uh, are reabsorbed in certain sections of your kidney. So the cells of the nephron will secrete hydrogen ion. So there's a neat secretion of your hydrogen ion to eliminate excess acids in the body into the tubular fluid. So when the blood goes through the glomerulus and is filtered through the glomerular capillary, so here you have filtration of blood, the, the tubular fluid will flow through the tubules. And during that, along the segments through which it flows, there occurs a reabsorption of the filter load of bicarbonate. So the contribution of each segment of the nephron to the process of reabsorption is shown here in this figure. So about 80%, no, of your filtered plasma bicarbonate is reabsorbed in the proximal tubule, and about 6% in the distal tubule, and about 10% in the thin ascending limb of the loop of Henle, and about 4% in the cortical collecting ducts. So that more or less all of the bicarbonate that are filtered in the glomerulus are reabsorbed along the length of your tubules. So at a plasma bicarbonate concentration of about 24 mEq per liter, and a GFR, no, or a glomerular filtration rate of about 180 liters per day, which is about average in a normal human being, the filtered load of bicarbonate is about 4,300 mEq per day. So this amount of bicarbonate that is filtered, uh, approximately 80% of this load, no, 80% of this filter load is reabsorbed by the proximal tubule, and 16% reabsorbed by the thick ascending limb on your distal convoluted tubule, and the remainder 4% is reabsorbed by your collecting duct. Now, let's take a closer look at the mechanism by which your bicarbonate is recycled in your tubules, and how it is able to eliminate hydrogen ion, no, excess acid from the body. So, the cellular mechanism by which hydrogen ion and bicarbonate are transported across the apical, across the apical and the basolateral membranes of the proximal tubules are illustrated in this schematic figure. So hydrogen ion secretion across, so this is the apical membrane. This is the tubular space. So this is where your filtrate goes through before they're eliminated via the urine. So hydrogen ion secretion across this apical membrane here occurs via two mechanisms. So that is through the sodium hydrogen antiporter or your sodium hydrogen exchanger, or some books call them your NHE3 exchanger, and your hydrogen ATPs. But however, the primary mechanism by which your hydrogen ion is secreted into the tubular fluid is via your hydrogen, your sodium hydrogen antiporter or your sodium hydrogen exchanger 3. So the estimate is that about two-thirds of proximal bicarbonate reabsorption occurs via this secretion by your NHE3 or your sodium hydrogen exchanger 3. Your hydrogen ATPs, on the other hand, provides another mechanism for your apical hydrogen ion secretion and is responsible for approximately one-third of your bicarbonate reabsorption. So remember that uh, the purpose of these um, channels basically is to eliminate hydrogen ions, extrude hydrogen ion into the fluid, and at the same time to uh, recover your carbonic acid via this reaction through the carbonic anhydrase, where once your hydrogen ion is buffered by your bicarbonate in a reaction forming our carbonic acid, your carbonic anhydrase acts on your carbonic acid to produce your water and carbon dioxide. And since carbon dioxide can diffuse through the apical membrane, is reabsorbed into the cell. And once again, inside the cell, intracellularly, it can combine with water and via your carbonic anhydrase, form back your bicarbonate. So there is a net recycling of your bicarbonate, even though you are eliminating your hydrogen ion. Also, your hydrogen ion and bicarbonate secretion have another mechanism in the intercalated cells of the collecting duct. So remember, your collecting duct in the distal portion of your nephron are also responsible for hydrogen ion and bicarbonate transport. So here in this figure, you will see that you have another set of transporters, no, that allow for the elimination of hydrogen ion uh, out of the body. So your acid-secreting intercalated cells have bacular hydrogen ATPase, so your hydrogen ATPase, and you also have your hydrogen potassium ATPase. And both of them are localized along your apical membrane. And your bicarbonate exits the cell across the basolateral membrane in exchange for your chloride, this one here. So the less abundant bicarbonate secreting cell has a vacuolar hydrogen ATPase localized to the basolateral uh, membrane, and a different chloride bicarbonate antiporter called indrine in the apical membrane. So the basolateral membrane in exchange for chloride. So This two processes allows for the recycling of your bicarbonate and for the elimination of your hydrogen ions. Also, there is what is called a mechanism for the generation of new bicarbonate through the titration of urinary buffers like your titrable acids. So when the body is eliminating hydrogen ion and your bicarbonate can be depleted no, in the buffer because of the it tries to neutralize the hydrogen ion load in the body. There's a way for your bicarbonate to be produced. So when a secreted hydrogen ion combines with the urinary buffer, a new bicarbonate is generated within the cell, no? And eventually replaces the bicarbonate that was lost earlier in the titration of your non-volatile acids produced in the cellular metabolism to buffer your net indigenous acid production. Another scheme also for the elimination of your hydrogen ion is through the generation of your ammonia and the excretion of your ammonium, especially to deal with the metabolism of your glutamine. No? So an important aspect of renal acid-base physiology is the production of ammonia or what's called ammonia genesis and the excretion of your ammonium. So your glutamine in the kidney will have a net production of two molecules of your ammonium, and this ammonium will be um, utilized for as buffers, no, for your acids in the body. So the kidney takes glutamine and then metabolizes it to molecules each of your ammonium and your bicarbonate. So two molecules of ammonium and your bicarbonate. So the ammonium is excreted into the urine and your bicarbonate, which is the new bicarbonate, is returned to the blood where it replaces the bicarbonate lost earlier in the titration of your non-volatile acids or the buffering of your non-volatile acids. Also, this figure shows the what happens to your ammonium ions. That is the return to the blood rather than being excreted in the urine. So when this happens, the ammonium ion is converted to urea by the liver, and in that process, hydrogen hydrogen ion is generated. So this hydrogen ion is buffered by your bicarbonate, and thus will negate the process of the renal production of new bicarbonate. So that's from the perspective of your renal acid-base physiology. Ammonia production, ammonium ion production by the kidney must be excreted into the urine and not returned to the blood, so that the new bicarbonate that is produced will not uh, uh, be utilized for neutralizing the hydrogen ion that will be produced as a result of that. So for every milliequivalent of ammonia ion excreted, a milliequivalent of new bicarbonate is returned to the blood. So this process accounts for approximately two-thirds of your renal excretion of your acids. So let's continue to take a look at the renal handling of ammonium ion. So when your ammonium ion is produced from your glutamine, metabolism, what happens to the ammonium ion. So here's your glutamine, and then as a result of your glutamine metabolism, you produce ammonium ion, and of course also carbonate, based on our previous discussion. So the importance of the ammonium ion remaining inside your, being filtered in your tubular structures is that so that it will be able to preserve the bicarbonate that has been produced by your glutamine metabolism. So there are two mechanisms for the secretion of your ammonium ion by the collecting duct. So in this figure, you will see that there are two mechanisms. One which is a non-ionic diffusion and diffusion trapping of your ammonia. So you have a non-ionic diffusion trapping of your ammonia. Second is your here in figure B, the secretion of your ammonium ion via your RH glycoprotein, RHCG, or your RH glycoprotein. So in this figure, glutamine is metabolized by proximal tubule cells. And for each molecule of your glutamine metabolized, it produces two bicarbonate and two ammonium ion. And then the bicarbonate is returned to the blood as a newly generated bicarbonate. So it replenishes the stores of bicarbonate in the body that are used to buffer acid production in the body. And then your ammonium ion is secreted into the tubular fluid. So your ammonium ion is secreted into your tubular fluid via this exchanger here. And the majority of your uh, ammonium ion uh, secreted by your NHE3 with ammonium ion substituting for hydrogen ion on the transporter. So you have your NH80, your ammonium hydrogen exchanger. No? In addition, some of the ammonium ion may enter the tubular fluid as ammonia. So it can either go there as ammonium ion, or it can go into your fluid as in the form of ammonia, where it is then protonated. So it combines with the hydrogen ion in the fluid. So regardless of the mechanism, for every ammonium ion secreted into the tubular fluid, what is important to take note is that there is a new bicarbonate returned to the blood as a result. So in the thick ascending limb of the loop of Henle, significant amounts of your ammonium ion are reabsorbed. So therefore, a return into your circulation. So multiple routes exist for this reabsorption, including your substituting for potassium on the apical membrane, of your sodium potassium 2 chloride importer, and the movement of your ammonium ion through the paracellular pathway. So ammonium ion movement out of the cell across the basolateral membrane can occur via your potassium channels. And this reabsorbed ammonium ion accumulates in the renal medullary interstitium and of course will contribute to your osmotic pressure in this portion of the kidney. It is also important to remember that the kidney has several ways by which it responds to an increase in acid production of the body. So in the proximal tubule, what it does is it increases secretion of your hydrogen ion. So when there's cellular acidosis because of the accumulation of your hydrogen ion, it will be excreted, no, secreted into the proximal tubule fluid. So your hydrogen and bicarbonate transport are increased along the nephron. So this results from a decrease in the intracellular pH as well as the effects of your AT1 and cortisol. So both your 81, your cortisol uh, has the same effect as a decrease in your intracellular pH, which means that there's an increase in hydrogen ion concentration. So ammoniogenesis, which is one of the responses of the kidney, and collecting that secretion of your uh, ammonium are stimulated. So we know that your ammonium ion is also one of the buffers, no, for your acid production. Finally, the proximal tubule phosphate reabsorption is inhibited. So there's inhibition of your phosphate reabsorption along the proximal tubule, allowing for the great excretion of your tritable acid. So when there's decreased reabsorption of your phosphate, it allows for excretion of more titratable acid in the urine. So the net effect is an increased renal net acid excretion, or your RNEA, no? Renal net acid excretion of the body. So there will be increased um, urine elimination by ammonium ion, um, increase urine presence of your tetrable acids, and of course, because your buffers are used up, there will be decreased bicarbonate, and bicarbonate will be reabsorbed along your tubules. At the distal portion of your tubules, there is also increased secretion of your hydrogen ion, the same factors that are, stimulatory in the proximal tubule. However, there is increased production of your bicarbonate in the distal tubules, and of course, this will allow you to be able to buffer tetrable acids, and of course, increase ammonium secretion by your RH glycoprotein. So that is how your kidney handles the acid load in the body and increases or decreases. So acidosis or alkalosis will lead to changes in the handling of your hydrogen ion and production of your bicarbonate and ammonium ion, your buffers, no, your urinary buffers along your uh, uh, nephron, along the tubules. So just to review, also clinically, life is only compatible between a pH of 7.8 and 6.8. So you know that the normal pH in the body ranges from about above 7.35 and below 7.44. So it's a very narrow range, no? And the extremes of this, it could reach up to 7.8 or as low as 6.8. However, beyond these figures, extremes of pH is no longer compatible with life. So therefore, it's important to measure pH, carbon dioxide, and oxygen in your blood samples to be able to see the clinical condition of your patient and understand what is happening in terms of your hydrogen ion concentration in the blood. Also, if you remember your Henderson-Hasselbalch equation, you can use that equation to calculate your bicarbonate concentration uh, through this the ratio between your bases and your acids in the blood. Also, laboratory measures total carbon dioxide concentration, your dissolved carbon dioxide plus bicarbonate concentration. And usually this is about 25 to 26 megs per liter in venous samples of blood. Also, laboratories measure total carbon dioxide concentration, and they exceed plasma bicarbonate concentration by about 1.0 to 1.5 megs per liter. And your normal plasma bicarbonate concentration is only about 24 mec per liter. So your carbon dioxide concentration is a little bit higher compared to your plasma bicarbonate concentration by about 1 to 1.5 mg per liter, because your normal plasma bicarbonate concentration is approximately 24 mg per liter. So when you have conditions of increased hydrogen ion concentration in the body, the net result is the reduction of the pH. So this is your acidosis or condition of acidemia. When there is decreased amount of hydrogen ion in the body. This will lead to increased pH, and this is a condition called alkalosis or alkalemia. So processes that lowers your pH will be referred to as acidosis. So whether it's metabolic processes or due to respiratory processes where you have increased production of carbon dioxide that lowers pH, it is called acidosis. However, processes that increase your pH, it's called alkalosis, whether it's metabolic alkalosis or respiratory alkalosis. Also remember, I showed this uh, equation earlier between your carbon dioxide plus water. So when your carbon dioxide is dissolved in the blood, it mixes with your water to produce your carbonic acid. And carbonic acid production of your carbonic anhydrase will produce your hydrogen ion plus bicarbonate. And it's important to note that your bicarbonate is a urinary, is a buffer of your acid, and it is used to react, no, to neutralize increased acid secretion or production in the body. So if it's in a closed system, the pKa is about 6.1, and the normal pH is about 7.4. However, because we are an open system where we are continuously excreting carbon dioxide through the lungs, this makes this system eliminating excess acid very efficient. No? So this makes carbon dioxide an efficient buffer because you can eliminate it via your lungs. So even if you produce about 15,000 millimole per day of carbon dioxide, it is easily eliminated through your respiratory system. So your determinant of pH, of course, will be dependent on the ratio of your carbon dioxide to your bicarbonate, and pH can be converted to your hydrogen ion concentration. So pH of 7.4 is equivalent to about 40 nex per liter. And so when you have 7.4, 40 nex per liter, so if you have 24, if you have concentration of 24 mex per liter, your carbon dioxide, let's say, is 40 and your bicarbs 24, that will be your ratio, no, of your pH. So the normal pH is, as I mentioned earlier, is 7.95 to 7.5, and you can uh, compute your pH by an inverse log of your hydrogen ion concentration. So if your hydrogen ion concentration, you can compute it by multiplying 24 times the partial pressure of your carbon dioxide in the blood divided by the concentration of your bicarbonate. So this could be, that could be your computation here. If you have about 40 partial pressure carbon dioxide in the blood divided by 24 it's per liter of your bicarb in the body, in the blood. So your normal pressure of your carbon dioxide in the blood is about 40 millimeter mercury. So it ranges from 36 to 44 millimeter mercury. And your normal bicarbonate concentration in the blood is 20 to 26 mg per liter, or about 24 mg per liter. So the ratio is about 40 to 24 mg per liter ratio between your carbon dioxide and your bicarbonate. So also remember that processes that directly alter bicarbonate concentration uh, will lead to metabolic disorders. No, so metabolic acidosis is due to decreased bicarbonate in the body. Metabolic alkalosis results from increased bicarb, no, in the body. On the other hand, respiratory disorders are those processes that directly alter carbon dioxide. So you will have respiratory acidosis if you have increased carbon dioxide, for example, in patients who have problems breathing, so that they are able to eliminate carbon dioxide from the body. Or those who are hyperventilating can result to respiratory alkalosis. You have decreased carbon dioxide. This can also result to respiratory disorder. So there's a buffering effect by slightly increased bicarbonate with respiratory acidosis. So your body tries to compensate uh, to your respiratory acidosis by a slight increase in your bicarbonate. And also there's a slight decrease in your bicarbonate with respiratory alkalosis. Remember, your bicarbonate are your buffers, no, yeah, for your acids. So buffering system works by preventing wide changes in the pH in the blood in response to the addition of a base or acid, because we know that the normal physiologic pH is within a very narrow range, no, that is uh, acceptable to life. If the extremes of pH, it will lead to uh, disorders that could be fatal. So buffering system using your bicarbonate and your other urinary buffers are actually important to in order to deal with changes in pH as a response to increased load of acid or bases in the body. So bicarbonate is the major extracellular buffer and it can be easily measured, but there are also intracellular buffers like phosphates, etc. So the buffers I mentioned earlier allows you to deal with indigenous production or iatrogenic intake of your acids or bases. And usually these are like emergency systems that can immediately neutralize. However, buffers are limited because they are not they are not that let's say they can be depleted, no, by extreme conditions of acidosis or alkalosis. So these are some of the buffer systems that work in the body. So you have your bicarbonate buffer. You have your bicarbonate, which is the hydrogen acceptor, and your carbonic acid, which is your hydrogen donor. So this is your acid, and this is your base because it's your acceptor. Uh, Phosphate in the urine. So you have your phosphoric acid, and of course your dibasic phosphate. You have your ammonia in the urine also. You have your ammonia and your ammonium ion, which are uh, as your acid, and your protein also can actually act as buffers. Proteins are actually hydrogen acceptors and are also donors. So it's important to note that your proteins can easily be affected by excess hydrogen ions because they interact with hydrogen ion. That is why when there is so much hydrogen ion in the body, it will lead to derangement in the function of the body because of the nature of the protein hydrogen ion interaction. There are also secondary compensatory mechanisms in addition to your buffering mechanism. These compensatory mechanisms occur in response to changes in pH. However, they are present in simple acid-basic disorders. If they are not present, if the secondary compensatory mechanisms are not present, then it is a mixed disorder. So your compensatory mechanisms include your respiratory system, which compensates for metabolic disorders, the altering carbon dioxide via the lungs, which is rapid in onset and many minutes, and compensation for your respiratory disorders occur by alterations in bicarbonate concentrations by the kidney, which is slower in onset of 1 to 2 days. So these are the other mechanisms that play in the buffering of an acid load. You have your increased rate of depth of breathing, your buffer systems like your phosphate and your protein. You also have increased hydrogen ion excretion in your kidneys, etc. So in summary, your conditions of metabolic acidosis is associated with decrease in pH, and the response will be the buffering by your bicarbonates. There will be a decrease in your bicarbonate in the blood, and a compensatory mechanism is an increase in your carbon dioxide elimination, so it will be decrease in carbon dioxide pressure in the blood, and all three markers go in the same direction. So in metabolic acidosis, it is uh, supported by the presence of low pH, low bicarbs, and low partial pressure carbon dioxide. In metabolic alkalosis, the opposite happens. There's increased pH, there's increased bicarb production, and there's also increased compensatory elimination of your increased partial pressure of your carbon dioxide in. So there is increased partial pressure carbon dioxide. So these markers go in the same direction. Your respiratory acidosis, on the other hand, you have decreased pH, but your bicarbs are increased and your carbon dioxide are also increased. So pH goes in opposite direction to the other two markers. In respiratory alkalosis, you have increased pH. You also have decreased bicarb and decreased carbon dioxide. Your pH goes also in opposite direction as your other markers. So you see that in respiratory disorders, the pH is in opposite direction to your two other markers, while in metabolic and disorders, either acidosis or alkalosis, all three markers go in the same direction. So these are your golden rules for your simple acid-base disorders. CO2 and bicarbonate always change in the same direction. Secondary physiologic compensatory mechanisms must be present, so your respiratory changes must be present, and the compensatory mechanisms never fully correct your pH. So these are the processes that causes metabolic acidosis, decreased renal acid excretion, direct bicarbonate losses, increased acid generation, etc. And this is listed in one of your textbooks where you have listed down injections of substances like aspirin, ethanol, methanol can lead to acidosis, or your lactic acidosis, or your ketoacidosis diabetes can also lead to increased acidosis, or loss of bicarb like in diarrhea, in the GI tract, or pancreatic biliary intestinal fistulas, or if you have a type 2 renal tubular acidosis can also lead to loss of bicarbonate leading to metabolic acidosis, or you have decreased acid excretion. If you have renal failure, decreased excretion, type 1 distal renal tubular acidosis, and type 4 renal tubular acidosis, hypoaldosteronism can also lead to metabolic acidosis. So respiratory acidosis, on the other hand, can be induced by hypercapnia or decreased alveolar concentration, ventilation, and buffering mechanisms to raise your plasma bicarb concentration. It's a rapid but limited response, and your kidney minimizes the change in your extracellular pH by increasing acid excretion of ammonium generating new bicarbonate ions. So this is one of your tables where this compares the different types of renal tubular acidosis. You have your type 1, which is a distal tubule type of renal tubular acidosis. Type 2 is proximal, and you have type 4. Just take a look at this in your textbook for more explanations. So on the other hand, your respiratory alkalosis is due to reduced carbon dioxide due to increased alveolar ventilation, etc. And your metabolic alkalosis are processes that raise plasma bicarbonate concentration, including the loss of hydrogen ion from the GI tract, which I had mentioned earlier, and your excessive urinary acid excretion. So major causes of your metabolic alkalosis will include hydrogen loss, administration of bicarbonate or organic ion that can be metabolized to bicarbonate, and what is called contraction alkalosis due to your loop or thiazide type diuretics in edematous patient, or vomiting or NGT suction in acromegalia, or sweat losses in cystic fibrosis. So for your metabolic alkalosis, your urine chloride concentration may be less than 20 mg per liter, but it can also be less than 10 mg per liter in chloride responsive. But in chloride resistant, your urine chloride can be greater than 20 mg per liter, and it is usually 50 mg per liter. So these are the expected changes in your pH for your respiratory disorders. So these are the ratio of changes in terms of your concentration of your bicarbonate in your carbon dioxide in relation to the four respiratory disorders and um, acute and chronic acidosis and acute and chronic alkalosis. So these are your primary and compensatory changes in the different acid-base disorders. In metabolic acidosis, you have fall in plasma bicarbonate concentration, and the compensatory response is reduction of partial pressure carbon dioxide averaging about 1.2 millimeter mercury per 1 mg per liter reduction of plasma bicarb concentration. In your metabolic alkalosis, you have a rise in plasma bicarb concentration, elevation in your carbon dioxide is the compensatory mechanism, averaging about zero point 0.6 to zero point 0.7 millimeter mercury per 1 mg per liter rise in plasma bicarb concentration. On the other hand, your respiratory acidosis leads to elevation in your carbon dioxide. And if it is in acute compensatory mechanism, you have a rise in plasma bicarb concentration averaging about 1 mg per liter per 10 millimeter mercury elevation in PCO2. If it is chronic type of condition, then your increase in plasma bicarb concentration average is about 3.5 mg per liter per 10 millimeter mercury rise in PCO2. On the other hand, your respiratory alkalosis, there's a reduction in carbon dioxide. So the compensatory mechanism, if it's an acute disorder, there's a fall in plasma bicarb concentration averaging about 2 mg per liter per 10 millimeter mercury decline in PCO2. If it's chronic, the fall in plasma bicarb concentration averages about 4 mg per liter per 10 millimeter mercury decline in PCO. So in our next discussions, we'll be taking a look at how you can how do you approach patients presenting with different types of disorders uh, in the renal system, including this acid-base disorder. So these are just a review of what we have discussed earlier. This is schematic diagrams of the secretion of your hydrogen ion in the intercalated cell of the cortical collecting duct, secretion of bicarb in the intercalated cell of your cortical collecting duct, transport of sodium in the principal cells of the cortical collecting duct. Sometimes also you're asked to compute for plasma and ion gap. So just review uh, the strong acid dissociation, physiologic pH, and how they are buffered in the urine and how they are computed. We don't have time to discuss this. Just review the physiology of that. So sometimes also your titratable acid are asking questions. So titratable acid basically is a way to compute for how much you need to, how much acid need to be neutralized that are present in the urine. So it's measured through your titratable acidity. Finally, take note that there are also what is called mixed acid-base disorders, where the presence of more than one simple acid-base disorder is noted simultaneously. For example, you can have respiratory acidosis with metabolic acidosis. So it's both types of acidosis is present, respiratory and metabolic. So this is called profound acidemia. You also have respiratory alkalosis plus metabolic alkalosis, which is profound alkalemia. And you also have metabolic alkalosis and respiratory acidosis, or metabolic acidosis and respiratory alkalosis. So we will go to a discussion of your approaches to patients presenting with various disorders in your next session.